Welcome back. We'll see biotechnology in agriculture. In that first one, we'll see pest resistant plants. Okay, pest resistant plants. So here, what is the traditional technique? Traditional technique, we used to make hybrid only, but we used to spray pesticides over a plant, or we used to spray insecticides over a plant to safeguard the plants from pest or to safeguard the plants from insects. But it is a time consuming, and also it is a economically it is very economically, means we want to spend more amount every time we want to spray pesticides, and also we need to spray chemicals. To overcome this problem, now scientists they designed, now biotechnologists they designed GMO plants, genetically modified plants. So in that first one we can see pest resistant plants. So here for this plants, for some of the plants, and what I'm going to explain example, no, in that they'll resist against pests. So we don't want to spray pesticides, or we don't want to spray pesticides. So how the plant will attain pest resistant character, that is only GMO technique, that is only we are going to learn in this class. Now look at the slide. Development of pest resistant plants reduce the use of pesticides, because we already changed the gene, so automatically we don't want to use the pesticides. And one such approach, introduction of Bt toxin to target plant. Examples: Bt cotton, Bt corn, rice, tomato, potato and soybean. These are the some plants or GMO plants, genetically modified organism. They will resist over a particular pest. They'll resist over particular pest. So we are going to learn in detail regarding the Bt cotton. So first one, Bt toxin. So what do you mean by Bt toxin? It is Bt toxin means expansion for Bt is Bacillus Thuringiensis. It is a bacterium, and it will possess a Bt toxin gene. Bt toxin gene. What is the gene name? It was given in your book. CRYIAC. CRYIAC. That is gene. And what it will produce a protein? Cry protein or Bt toxin protein. So gene CRYIAC, and it will produce a protein called cry protein. Clear, children? So sometimes when we write in italics, that is a gene. When we write in normal letter, that is a protein. When we write in italics, that is a gene. When we write in a normal, that is a protein. Now Bt toxin, look at the slide. Bt toxin gene cloned from bacteria and expressed in plants to provide resistance to insects. No need of insecticide here, or that is we can call it as biopesticide. So we are taking Bt gene from Bacillus Thuringiensis bacterium, and we cloned in some other bacterium, and we introduced into plants like cotton plant, and the cotton plant will have the resistant power to resist against some of the pest. So how all will happen, that is only we are going to see here. Okay, we'll see. So how the mechanism of Bt toxin and Bt cotton, we'll see in detail. Okay, so Bacillus thuringiensis, so we should not underline us together. Bacillus thuringiensis. Bacillus thuringiensis is a bacterium, and they will produce a one toxin protein, Bt toxin, that is cry protein, which kills insects like lepidopterans, that is tobacco buttworm or armyworm. This lepidopterans includes butterflies and also moths. And here buttworm and armyworm, actually whatever the tobacco buttworm or armyworm, everything in their larval stage only, not in the matured stage. We know the life cycle of butterflies, or first it will lay egg, second larva, third one pupa, fourth one butterfly. So when larva, when it used to eat the Bt cotton, what will happen only we are going to see. So in the larval stage even, this compound will work. It is not in the mature stage of butterfly, or it is not in the pupa stage. Whatever we are going to talk, everything in the larva stage of the worms, like butworm. Tobacco butworm means it will affect very particularly tobacco plant, and armyworm also, everything in the larva stage. And coleopterans is, we know, beetles, what we learn in 10th standard, red beetles and other things. And dipterans means we can tell actually these organisms, they have two wings in their body, so that we can call it as dipterans. For example, organisms like flies and mosquitoes, organisms whose have two wings, we can put under the category of dipterans, that is flies and mosquitoes. To escape from lepidodendrons, lepidotropins, not lepidodendron, that is pteridophytes, okay. Here you want to remember lepidopterans, coleopterans and dipterans. So to escape from these kind of pests or insects, how the Bt toxin is working, only we are going to see. Is that clear, children? So Bt toxin gene express itself a protein crystals at a particular phase of bacteria, that we can call it as insecticidal protein. Okay, Bt toxin exists in inactive prototoxin. Okay, normally the Bt toxins will be produced by the Bacillus Thuringiensis, children, and it started to produce Bt toxin. This Bt toxin, it will be in inactive stage in bacterium also. It will be inactive stage in in cotton plant also. But it was eaten by, suppose it was eaten by the larval stage of the organism, means this Bt toxin will get into their body, and the body pH, because we know organisms in that normally in human digestive system, even if pH alkaline only, it will activate some protein digestion. So what we learned in our human digestive system, alkaline will initiate the pepsin, so pepsin will help to break proteins into amino acids. Likewise, what is Bt toxin which is present in bacterium also, they never contain the situation to break. So it is not harmful for bacterium also, and it is not harmful for plant also, because the alkaline pH won't be in plant, but it was invaded by these kind of worms in their larval stage, and whenever the toxin material will get into their body, automatically the alkaline condition of their cells will makes it into active form. So once it was makes it into active form, automatically it started to convert into active form, and you will get a crystalline proteins, okay, that is soluble. The crystalline proteins will get dissolved in the alkaline pH, and once it dissolved, automatically what will happen, it started to make small pores in the epithelial cells of the gut of larva. It started to make pores in the epithelial cells of the gut in the larval stage. Once it started to make pores, automatically there we can see, because from extracellular fluid also everything will get inside the cell, and you will get swelling of cells would occur, and also leads to one important process, we can call it as paralyzing of a cell would occur. What do you mean by paralyzing of would occur? Paralyzing means it won't work properly, so it is enabled to do its own function. So the epithelial cells of gut cells, they enable to do its own work, so automatically the epithelial cells started to die. If digestive tract is started to die, or it can't able to do its work, and automatically the larva stage, even the organisms will die. So that is the basic principles behind Bt toxin. And also when we introduced in different kinds of plants, it is not a problem for that plant, because it is inactive. The Bt toxin will be inactive in plants, so it never disturb any kind in plants, but it was invaded by the organisms, it gets activated, it will disturb the gut cells of the epithelial cells of larva, and it gets swelled and automatically leads to death of the cells. So many cells were died, automatically leads to the disturbance in the digestive system and leads to the death of the particular organisms. So automatically it is no need to spread extra insecticide or pesticides to kill larva when it started to have the, because it will be, the Bt toxin will be present all over the parts of the plant. It is not only present in leaf or stem, because we already introduced in gene even. So wherever the gene will be present in throughout the body system in plants. So which part of the plant it was eaten by the larva will die because of the activated Bt toxin in their body. So the larva will die. Okay. So, and what is the gene responsible for that? It is CRYIAC, and it was isolated from the bacterium, and it was okay. Now look at the slide. So activated, that story already we learned. So Bt toxin isolated from bacteria Bacillus Thuringiensis, coded by gene CRY. Okay, CRY, that is IAC. What is given in your book? So CRY IAC, that gene, and different types also, and incorporate into crop plant depending on the type of crop and target pest. So CRYAC is a one gene, like that CRY2IAB for cotton bollworms and CRYAB for corn borer. So CRYIAC, and only one gene was given in your text, children. You write these three also. CRY1AC and CRY2AB, both for cotton bollworms. Okay. And CRYIAB for corn borer. Corn borer means normal your corn only, maize only. Okay. So these are the genes responsible to make CRY proteins. It will be in inactive stage in bacterium also, when we introduced in plant also, but plant was eaten by the bollworms or like larval stage of all lepidopterans and coleopterans and diphterians. Automatically they will die because CRY proteins activated in the alkaline pH in their body. So automatically they die, children. Is that clear? So this is the process how we are introduced to the this gene. Okay, CRYIAC. Now look at the slide. Here the bacterium has the flagella. What we used to see, Escherichia coli, they doesn't contain any flagellum, but here we learned in lower classes, now 11th standard, they have atrichus, bitrichus, like that. Based on the flagella we have a type. So here monotrichus, not monotrichus. All many flagella will be in the one group of the bacteria. Sorry, one bacteria we can see flagella in one side. So automatically many flagella will be in the one side of the bacterium. So we are isolating the DNA. How we can able to isolating the DNA from that? We are introducing lysozyme enzyme to kill the cell wall of the bacterium, and we are using other enzymes to break the nucleic acid. From that we are getting DNA. So DNA cloning, and the DNA, what is our desired part, we should found, and we should cloned in the other bacterium. And after cloning, we should transformed into the Petri plates, that is transformant containing recombinant DNA, and we should culture in the Petri plate. We should culture in the Petri plate, and this Petri plate, from that we need to introduce into the plant. Okay, we need to introduce in the plant using biolistic, that is normal. No, from that gene gun we can introduce into the plant. So now the plant will possess the characteristic of the Bt toxin characteristic. So automatically when it start, now look at these worms. Okay, when this started to have the corn, this larval stage, even it will die because of the Bt toxin, and even this even flower bud, even when it is have, no, because I told you Bt toxins will present in throughout the parts of the plant. So which part it will invade also, it will die. So normally in those days they used to give some information regarding not to have GMO crops, because it will cause impotency to male also, female also, because once when we have, even human beings will have the alkaline conditions to digest, and it will make some toxins in our body also. So they told not to have the GMO crops. So many problems occurred in those days to have GMO or should not take a GMO crops like that, and what to do at last we should follow. Okay, next slide I'll explain this one, children. Thank you.